Good evening, everyone, and welcome to the York Festival of Ideas. My name is Jack Hatfield. I'm a postdoctoral research associate at the Leverhulme Centre for Anthropocene Biodiversity here at the University of York in the UK. And I'm going to be chairing this event this evening. So thank you all for joining us. It's my great pleasure to welcome you all here. So before I go on to introduce you to the speaker, I just need to run through a few technical notes here. Um, so subtitles are available in this event. To turn these on or off, use the CC Show Captions button at the bottom of your screen. If you are watching this event live, you can ask questions using the Q&A button on your screen. This is available throughout the event, so questions can be posted at any time. Please also remember that today's event is being recorded, so you'll be able to watch it again later. And now it's my great pleasure to introduce you to our speaker this evening, Karen Lloyd. Karen is joining us this evening from the US, where she is the Wrigley Chair in Environmental Studies and Professor of Earth Sciences at the University of Southern California. Her research focuses on microbes that live in environments such as the bottom of the ocean, hot springs, or under Arctic permafrost. Her work has appeared in leading publications such as Nature and Science. And this evening, Karen is going to talk to us about the strangest life on Earth. So that's over to you, Karen.
I basically want to talk to you about my obsession with life inside Earth. I want to know what's down there, and I want to know what it's like and what it's doing and how it's different from what we see up in the surface world. And so this is what I've spent my entire career working on. So I'll share a little bit of that with you. In the background of this opening slide, I have volcanoes in the Andes, which is leading into my I'm going to start by talking about some volcanoes because, we don't think about volcanoes as being hospitable for life. But when you want to study life that's deeply buried inside Earth, something that moves vast quantities of the Earth's crust back up to the surface is a good place to look for it. So volcanoes are really powerful places to look for that interchange between the surface world and the subsurface world. So this is a picture of Poas volcano in Costa Rica from the visitor center. This is a very touristy volcano because it's a beautiful volcano. And there's a lake in the middle of it that is full of basically battery acid. There's a bunch of reduced sulfur compounds that come out through the volcano, and they interact with water to form very, very acidic material. And this red section right here is basically the throat of the volcano. This is, if it erupts, this is where all the material comes up. But you can see it looks pretty calm, looks pretty good this day. So that was the day that I went into it. So this is me, the second person here in the hard hat. We wear hard hats when we go into the volcano because it's not a good place for hiking. It's not open to the public. I was going simply because I'm with my colleagues from Costa Rica who monitor this volcano, and they need to go inside it every now and then to check the gases. So we were going there as scientists with them. And so you can see the tourists here at the visitor center as we're going down. But you wear the hard hat in case any of the rocks around you come tumbling down. If it erupts, the hard hat's not really going to do much for you. So here's another picture of us hiking down into the crater of the volcano. Do you see us? I'll give you a second to spot us. It's like a little find it thing. There we are. So we're walking on the little pathway. We had a camera crew with us, and we offered the possibility, or no, I think we said, I think we're not going to allow you to come into the volcano with us. And they said, oh, darn. Oh, well, just got to stay and use the telephoto. But it was kind of an arduous hike, just a few hours. But you can see that we're nearing that big acid lake. When we got up close to it, you can actually see the volcanic gases coming out of little holes in the rock. And it forms these, there's so much sulfur in the gases, it comes out as sulfur dioxide, that it forms these beautiful crystals of pure elemental sulfur that give the thing these yellow colors. And this is just, you can almost watch them precipitating before your eyes. It's absolutely stunning to see Earth be so powerful. So these are pictures of me, probably the most terrified I've ever been in my life. You can kind of see me gritting my teeth together. This is my colleague Donato Giovanelli from Italy, and these are my colleagues from Costa Rica and Woods Hole Oceanographic Institution here. And this is me. So you can see the angle that the side of this crater is, and it's not, nobody made a path through this. So it's all like crumbly, gravelly ejecta, basically. And so you have to like really maintain your footing, because if you fall, you'll slip into this acid lake that's pH 0.9, what I measure 0.85 that day. But the thing is, you can't steady yourself by touching the ground, because the ground is 100 degrees Celsius. So that's why I was so terrified. And you can see all these gases around. But we ended up sampling it without any ill effects, and everything went great. But then about a month and a half later, it's not going. Oh, that's weird. I don't know why it's not going. Oh, that's too bad. I'm sorry. I've never had this not work before. Oh, there it goes. It's like it's not loaded or anything. All right, well, we're gonna watch it not as a video. I'm just gonna click through. See the little explosion? See it getting bigger? So this is basically, actually, it's a little more terrifying with like the stop motion version, because it happens really quickly if you watch it in video format. But that's where I was standing. So I was right there holding on to the side of the wall when you saw me last. And so this explosion just takes over and it comes out. And the deal is that it is basically like a mountaintop that just kind of blows its stack every now and then. And this ended up being the biggest eruption that they'd had in 60 years. And, I don't want to overemphasize the sort of cowboy nature of this thing that we do to get these precious samples. I mean, you can see here on this day, there's a lot more steam coming off this. There had been little pops, little phreatic eruptions, small ignitions every once in a while. So this was the day that this eruption happened, was not a day that I would have ever been in that volcano. So, it's a little bit of a mitigated risk, but it's still a risk. So the question that we want to ask with our research is, how can anything live in battery acid that explodes all the time? It just seems like a terrible place to live. So what we've learned, and this is not just my research, this is from the general field of acidophile research. So acidophiles are things that prefer acid. So to understand how they deal with acid, we need to talk about how all cells stay alive. So they basically move protons, so that's a positively charged hydrogen atom, outside the cell. We're doing it right now. All of our cells are currently pumping protons outside of our cells. And the reason that we do that is to set up a gradient. We set up more protons outside our cells than inside our cells. You can see there's more little H pluses outside than there are inside, my little fake cell that I drew here. And the reason that we do that is so that they will naturally want to fall back in. And when they come back inside the cell, it's like a waterfall. And so you know how you can like hook up a grain mill to a waterfall and use that free energy of falling water to spin something big and grind grain or mill wood or whatever you want to do with a waterfall. We inside our cells chemically do the same thing with this proton gradient, and we use it to make ATP, which is one of the common energy currencies of the cell. We do other things with it as well. So now, okay, that's normal life. That's everything. Let's go back to the acid lake. Acid is low pH. So pH 2, pH 1, those are extremely low pHs, and that's acid. Acid, the pH value, the equation that you use to get that is minus log of the protons. So the concentration, the amount of these protons, these H pluses that you have, determines how acidic you are. So the situation that we have, that we want, that the cell wants, is more acid outside the cell than inside the cell, and then we can get that nice gradient set up. But when you go to Poas volcano, the situation is that there's massive amounts of protons outside. So now it's like too much of a good thing. It's like all of these protons are sitting outside the cell, and now you've got a gigantic waterfall rushing inside the cell, which should be great. Like it should basically be free energy. The cells should just be able to sit there and just kind of like make ATP for free all the time. But the problem is that it's like drinking from a fire hose. You can't actually survive with that much energy like coming at you. So the way these cells deal with it is that they toughen their membranes to withstand this crush, and they add positive charges on the inside to decrease the gradient. But at the end of the day, sometimes they just have to bail. So it's this weird like mixture of good and bad to be living in an acid lake, because you get all these protons coming in for free, but it's really more than you want. So you've got to toss some out to handle it. But it seems to work. We find microbes present in all kinds of volcanoes. They are not present in lava, but this is a volcano that, oh, so sad. I gotta look into why my videos are not videoing. But we'll do this one stop motion as well. But this is Fagradalsfjall volcano that erupted recently in Iceland. And there's nothing, as far as we know, alive in lava. But what this did was this made changes in the area around it. And so it created hot springs around in the surrounding area and fed them with extra volcanic gases from deep inside Earth. And we were able to look at those and the effects of this giant volcano popping up in the middle of Iceland on everything around it. But it's the most spectacular volcano I've ever seen. And as we were going in to study it, we kept running into geologists in the airport. Pretty much everybody went to Iceland to look at that one, because if you study volcanoes, you're definitely a volcano enthusiast, and sometimes you'll just have to go run and check one out if it comes up. So this is an idea of the total landscape that this volcano encompassed. So this is me in the walk up to that volcano. You can see the volcano erupting in the background here. So I was obviously a lot closer when I took that video before. That was not a telephoto. That was just from my phone. But this stuff right here is fresh lava that this volcano just made. And so this is the advancing front. This is not cold, dead rock. This is hot rock, and there's lava underneath it. But it was slow enough moving that we could sort of sit around it. So basically, if you knock, this is me taking some of the lava out from my colleagues who are studying the crystals inside the lava. The outside of this lava cools and forms rock, but if you chip that away, if you knock that away, then underneath it is lava. So I'm not painting a very pretty picture of living in a volcano. It seems like a really bad choice for life to do. Why would you do this? And the reason is that volcanoes provide a lot of energy. One thing that we've learned when we study these strange microbes inside Earth is that energy is a fundamental rule or rubric for biology. It is the thing that is driving a lot of this life, and volcanoes provide a lot of energy. So one way to understand that is to compare it to what we know pretty well. So life at the surface of the Earth is really chemically boring. We do one thing, well, I guess technically two things if you include plants, but all of us animals, we use oxygen and we react it with carbon, with sugars and the food that we eat to make carbon dioxide. Puppies do it. Mosquitoes do it. Things that don't look like animals but are, like sponges, do it. And even these things that don't seem very closely related to puppies and sponges and mosquitoes, like amoeba, also only do that. So it's a very limited reaction that we do up at the surface world. And of course, plants also use photosynthesis to make oxygen and the organic food that we then can breathe, or that we can eat and then use oxygen. Okay, right, you know all that. But microbes inside Earth can use an almost infinite number of chemical equations to get energy. They're not limited to that oxygen equation. They can basically take the periodic table, and anything that can be reduced or oxidized within this periodic table, with a special emphasis on things right here in the middle, and also carbon, nitrogen, and sulfur are very productive elements to be used for this. Anything that can undergo reduction in oxidation chemistry will be used by some microbe somewhere. So there's one place on Earth that is probably better than any other place on Earth for finding this huge diversity of different things that can be breathed, and that's hydrothermal vents at the bottom of the ocean. So they kind of look like volcanoes, and sometimes people call them volcanoes in the ocean, but they're not volcanoes. They're actually water that has been sucked down slowly and superheated in the subsurface, and then it shoots out in these jets. And I don't know if you can see these things, but all these little red and white things around them are gigantic worms. And these are teeming life at the bottom of the ocean, which is not a place that people thought life was going to exist in any kind of big capacity, because we presumed up until about 1977 that most life was dependent upon sunlight. And so the first time people saw this temperature anomaly from these high temperature vents and went in a submersible down to see them, that must have been the most incredible feeling, to go down there and be like, what is down here? And to land and to see this teeming mass of biomass at the bottom of the ocean. People had no idea how powerful this is going to be, but it shows us that the chemistry that's from these deep subsurface sources is enough to really make life flourish. So one of the ways that we go and study these vents is that we go in this submersible called Alvin. This is the United States single deep diving submersible. We don't have any other ones. Some other countries have some deep diving submersibles too, and we collaborate. These are always international collaborations, so it's kind of nice. And it is dropped off the back, like placed into the water off the back of a support ship called the Atlantis, a research vessel. So this is what it looks like when we're launching. This is on the deck of the ship. We have a little train track to pull the submersible back into its little hangar. And we set up this front, we call it the basket, to do all the work with. And you can see the porthole right here. All of these people standing around are all pilots. So they are both the engineers that take care of the submersible and the pilots who drive it. They do both. And so the scientists, these are some scientists, these are actually some of my colleagues from Germany, are responsible for setting up the basket. So this is me setting up all night. We work setting up this basket and making sure that it's ready so that we can take cores, we can take samples for the next day's dive. And then the next morning, we launch. So notice this is myself and my advisor, Andreas Teska. We are not wearing shoes. You're not allowed to wear shoes in a submarine. What would be the point? And I'm not dressed very warmly right there, but I had lots of warm clothes sitting for me inside the submarine, or the submersible, because it is cold at the bottom of the ocean. It's very, very cold. So we were all bundled up. Here you can see me more bundled up here. This is what it looks like inside the submersible. And all these control panels are not exactly, they don't look like a Tesla. They're rocker switches, metal rocker switches that look like they're from the 60s, because they are from the 60s, because if it works, don't change it. So everything is redundant. There are analog versions of the support systems for the submarine and digital versions of the support system for the submarine. But there's not huge amounts of LED panels and stuff, because the number one fear in this submersible is actually fire. So you don't want a lot of really hefty electronics down there that could cause sparking. So everything is at a really low voltage. And I just love that. I love that disconnect between real ocean exploration and sort of how it's portrayed in the movies is like this high-tech, high-powered thing. And it's like, no, actually, it's quite a bit more practical just to use what is tried and true and is not going to fail and is not going to cause you problems. And it works just great. So it's wonderful. I have no worries going down in the submersible. This is what it looks like looking at a bacterial mat. So we see, again, this proliferation of life that we see at the seafloor extends to the bacterial biomass as well. So we see so much bacteria that it's visible to the naked eye. And these are these are not the kind of bacteria that would ever make you sick. These are tiny little filaments of sulfur using bacteria that are not all that different from what we find in volcanoes. I don't know if this video is going to work. Nope. But this is video that I made at the seafloor of these worms. You can see they have these heads, and so yellow feather heads. And so what they do is they sort of breeze around in the water, and they collect this vent water that comes up and feed it to the microbes that then make food for them, which is pretty cool way to live. And then we come back up, we bring all these weird, creepy, crawly things. You can see that we get really far away from the support ship. Divers help us before we launch, before we go down, but then they leave and they get in a little boat and they go back to the big boat, and then you're just untethered and working down there. So what we discovered during some of these dives, and this is kind of a big thing to say, but it is the truth, we discovered things that changed our conception of what types of life are on Earth, like what we know life is like on Earth. So this, up until recently, this was what we thought the tree of life was. And this was a new tree of life around 1990. We have us, the animals are all here, the plants are here, fungi, mushrooms are all here, and then these are mostly like single-celled type of organisms here. And we're all the eukaryotes. We have complicated cells, we're bigger, we're multicellular. We also have this other branch called the archaea, which are kind of like the bacteria, which are better known because this is where all the diseases are. This is where the pathogens are. Archaea are not known to be pathogens, but they're found in hydrothermal vents. They're found in high temperature places, but we've since discovered that they're found in other places as well. So after we came back from that cruise, and after my colleagues around the world came back from lots of other cruises, we discovered that the tree of life doesn't look like this. It looks like this. And this is not an exact representation, but basically we found all these new deep branches on the tree of life that no one had ever known before. So it meant that we are more diverse. We as life, not as a species, but all of us, there's a lot more diversity of life than we knew about before the early 2000s. And one of these types of life has now been gotten into culture, and this is really exciting. I did not do this work. This is some colleagues of mine in Japan and also in Austria independently got these in culture. And what these are the closest relatives of the eukaryotes. And so this may look like, okay, any other, you've seen lots of pictures of cells that have sort of arms that reach out. But what's interesting about these is that they're tiny. These are only they are the width of the wavelength of visible light. And so the fact that they have these structures, the fact that they're so tiny, and they're not eukaryotes, they're not amoeba, they're archaea, which are supposed to be simple. They're supposed to be prokaryotes that don't have really exciting cellular structures, but they do. These ones do. And they have these pseudopod-like extrusions that actually contain internal bits of the cell in them. It's something that we've never seen before in the prokaryotes. And that is a very hot area of research right now. And so I expect to find new stuff coming out, day by day in this work. So I'll wrap up with talking about just what life is like on the whole Earth and what it means for us as humans. So if we were live, I would ask you all to tell me what planet this is, but maybe you've stared at it enough now that you can shout at your computer screens what planet you think this is, and hopefully some of you will be fun and say Neptune, but most of you will get that this is Earth. We don't often look at Earth from this angle, but this is the Pacific Ocean. This is New Zealand. That's Hawaii. It really emphasizes how important our wild spaces are and our oceans are. And when we think about life inside Earth, we have to think about what's underneath our oceans. So we've discovered that there's roughly 10 to the 29 living microbial cells buried beneath Earth's seafloor, which is a third of the microbes on the planet. And it's roughly 10,000 times more than the number of stars in the universe. So this is a really literally astronomical number. So much of the life on Earth actually is inside Earth. So what we're chasing after is a very important ecosystem, and just in terms of the amount of space that it takes up on Earth. And as such, we need to know how it intersects with our own human lives, because it's not just out there doing something independently of us. What we do can actually affect it and vice versa. So one of the ways that I really want people to know about, because this is happening, and this is starting to happen in an accelerated pace. And so the more public awareness about it that we have, the more that maybe we can do this in a respectful and responsible way. So people are working on deep sea mining. We know a lot about mining on land. It's something that needs to be highly regulated. Otherwise, there's a lot of exploitation of ecological resources and of the people who are involved in the mining. But this is now being proposed to be occurring in the deep sea. And the way it works is that you've got this crawler basically hoovering up the top 10 centimeters of muck across huge swaths of the ocean, piping it up to a ship all the way up here. And the ship separates out precious metals and critical metals that can be used for batteries to help us free us from fossil fuels. So that's one thing that's often brought up, is that this is going to free us from fossil fuels. So maybe that'll be a good thing. But I guess the question is, can we get metals in another way? And then the remaining sediments, the leftover sediments, have got to be put back down. And if you put them in the middle of the ocean, they're going to kill all the fisheries. They're going to kill all the biodiversity. And that's something that's pretty well known. We cannot be dumping this in the upper ocean, because it really will blank out all that life. So it's got to be piped all the way back down. And then somehow this all has to be regulated and checked up on. And I worry about that, because I know how hard it is for us as researchers to go to the deep sea. It's just very hard to work in the deep sea. And so I worry about how you would enforce regulation about deep sea mining. But this is happening. Another thing that is happening that's maybe a bit more positive is carbon capture and storage. So one thing that, one of the problems that we have with climate change is that we have too much carbon in our atmosphere. And so we can go to point sources, such as industrial production of steel and carbonates or cements, and pull out the carbon dioxide and pump it underground or offshore underground. This is far from a settled technology, and I'm not here saying this is going to solve everything. It's pretty clear that even if it becomes useful and it's put to use, it will not solve everything. We have to decrease emissions. We have to find alternative fuels. We have to do lots of other things to deal with the effects of climate change. But I believe that removal and storage could be a part of that solution as well. And so both of these activities are going to affect this deep subsurface community. So since we just learned that they exist, we're going to need to be involved in these efforts going forward to make sure that we don't destroy these ecosystems before we have a chance to learn about them. And we also don't put all these materials down in a place where the microbes that are already there sort of send it right back up again, or really mess up our efforts and gum up our pipes and things like that, so that we can work together with them and not against them. So this is my talk today is just like a rapid fire introduction to a bunch of these different issues. If you find this interesting and you want to know more about it, I did just publish a book just a few weeks ago. Kirkus gave it a starred review and called it a glorious gallop through one of the last and possibly most important frontiers of science. I think that's what I intended it to be. I'm not primarily a writer, I'm a scientist, but I love to write. And what motivated me to write this book is the fact that I would walk around, I live in Los Angeles, and I walk around the streets thinking, I wish the people around me could know how diverse life is. They would be so pleased if they knew the role of energy and how there's beings inside Earth that push to the very boundaries of what we think life could do. So I decided to write a book to share it with anybody who wants to know those things. There's another crazy thing that they do that I didn't touch on in this talk so much, and that's that we think that they can live for very, very long amounts of time, which really throws your brain for a loop, possibly over geological time periods, which is crazy. I also want to acknowledge public funding of science, because this is something that's been I think not properly understood amongst the administration in the United States, which I realize most of you are probably not in the United States, but this is something that affects all of us, because work that has been funded by the United States has been good for other countries as well. I have received funding from these agencies, including the British Antarctic Survey. I'm collaborating with some folks in the UK, and they supported our work in the Arctic, which is really, really fantastic. But if we don't have public support of science, then we'll be really limited to what individual companies can fund, and their job is to make profit. So they don't want to fund really blue skies, big discovery research. They only want to like do the things that will have a guaranteed profit. So it's not a substitute for public funding of science. So hopefully, I'm hopeful that the U.S. government will remember that that's what has always made us great and keep it going. So I am happy to take questions from anybody, and thank you so much for listening.
Great. Thank you very much, Karen. So yeah, so if you can put any questions in the Q&A box. So I'll just start us off with a question while people are typing. I can see a few coming in now. But yeah, I just want to ask, the more you discover about the life that exists in these kind of very harsh and previously unknown environments, how does it make you feel? Like, does it make you feel hopeful? Because in your book, in the talk, you highlight a number of avenues that these life forms could be really useful to us and inspiring. Or as touched upon with the deep sea mining, does it kind of worry you that that there's whole ecosystems we know nothing about yet we're likely affecting them? Or a
Isn't
bit of
it
both, probably.
a bit of both? Yeah, yeah, yeah. I mean, isn't it a true saying that like to really care about something is to be in pain that it could get hurt? So, I mean, that's true of anything that you feel strongly about. But I think that, being afraid of bad things that would happen is not a reason, to not learn about things. So that's I'm mostly just focused on the joy of discovery. It's nice to work in an area where things are so little known. It means that big discoveries are always around the corner.
Yeah, it definitely does sound there's huge areas of discovery to be made there, when comparable to things that were happening decades, centuries ago in other fields. It's really impressive. So we've got a few questions coming in from the audience now. So so kind of the first one is asking. So say it's clear that you love your job, but what's your most exciting discovery to date?
Uh, that is a hard thing to answer. I think probably it's the part I didn't really talk about so much here, which is the idea that these things live for hundreds of thousands of years, which is definitely a working hypothesis. It's hard to prove that. But if we presume that they do, then, I like to run. And so I've spent long runs just like thinking in my brain, okay, what are the implications? What would your life be like if you lived for 100,000 years? And that's just been so enriching because it just it expands my view of what life is, which is fun.
Great, thanks. So we got another kind of related question here that asking, to what extent do you think yet undiscovered life on this planet could stretch our understanding of what is possible? And having questions, especially thinking about other planets as well.
Yeah, I think that this work is directly opening up possibilities for life elsewhere in the universe. I know that back in the earlier days of Mars exploration, the Viking mission was largely seen as a failure to find life. Which it's interesting going back and reading the original papers from there. It's like actually their experiments were really good and not conclusive. So it's kind of I'm not saying they found life, but I'm like actually I feel like those experiments need to be just rejiggered and tried again. But that their failure to find things that are easily identifiable as life on Mars sort of ended the interest in looking for life on Mars for a little while. And then the discovery of hydrothermal vents that I talked about in the late 70s is, I think, largely responsible for the fact that interest resurged and efforts were made to send the modern era of Mars exploration because it expanded our notion of where life can exist. And so as we continue to look and find life that pushes closer to the edges and the boundaries of what we think life is, then that pushes us more to the boundaries of where we can find life.
Yeah, there's it yeah, it should be a real boon for a lot of different fields. Just showing what can be discovered. So we've got another question here, probably quite a tricky one actually. how far down below the surface does life actually go?
I mean, it's not tricky because I can just say, I don't know. Does. But the deepest that we found life is about 8 kilometers. And I say about 8 kilometers because this was from some deep mines, and the mines themselves were 8 kilometers deep, which is a massive feat. I mean, it just 8 kilometers doesn't sound that far when we're talking about driving a car, but going into the earth is something really special. But then when you take fluids from the fractures in these rocks, it could have been coming from a little bit shallower, could be going from way deeper, like who knows? I mean, at some point, life as we know it definitely ceases. It's not going to go into the mantle, deeply into the mantle. 500 degrees is not a possibility for life, but there's a lot above that. There's a lot of crust above that.
Not until we discover the lava bacteria that you said.
When we discover the lava bacteria, that's going to be amazing.
Yeah. Um, yeah, this is all a question about what type of reproduction this extreme life uses.
So by and large, so the ones that are in the sort of high energy places like volcanoes, it looks pretty similar. It's pretty typical. Just as long as you can like push the protons out, deal with the temperature, all the stresses of the environment, then they just divide. And some of them grow really quickly too. So they're actually nice to work with in lab because you make up. I don't know if you've ever grown bacteria in a laboratory, but you work really hard to make sure you're not contaminating anything and it's really sterile. So you're kind of treating these samples as like very, very gently. And it's funny growing extremophiles because you do the same thing to prepare them and you're taking such care. And then you literally throw them in a vat of boiling water. It's like, it just seems like you're going to kill them. But then, they grow so fast that you can come back the next day and you've got this like teeming mass of microbes that are really excited about that, boiling water you threw them into. So yeah, pretty typical for some of them. The really slow ones, I have no idea how they divide. It is something that I think a lot about.
Yeah, might take a few epochs to discover that one.
Yeah, I always wonder, are they like, do they do for DNA replication, which you have to, make a copy of your genome before you can divide into two cells? That's a pretty typical thing of everything. Are they like making one copy of a nucleotide per century? Like next century you get a G, next century you get a C, probably not, because
Well, yeah, I mean, at least if it takes thousands of years for them to reproduce, you can quite easily speculate, and it'll take a very long while for anyone to prove you wrong, right? So, um, so I have another question asking about what do you think the problem with why do you think the problem with deep sea mining is not higher up the agenda? It's not something that most people really hear about. Well, obviously from your talk, the consequences sound like they could be devastating.
they could absolutely be devastating. I mean, that's why I'm talking about it. I think that it's just an attention span thing. We have a lot of important things to pay attention to all the time. Human problems that we shouldn't ignore those things. So things that are in the deep ocean kind of happen without our notice, and that is one of my worries. If it is happening, and if it becomes economically viable and lots of people start doing it, then we really need to pay attention. It's not happening in the complete dark. The United Nations has the International Seabed Authority, which is regulating the early attempts, but individual countries can choose to do this within their own 200 miles offshore of their borders, in their exclusive economic zones, and that can happen as unregulated or regulated as a country chooses.
Yeah, I think it's probably a case of, I mean, it's the legislation around the oceans is quite complicated, even for some of the perhaps things that people do interact with, and such as fishing and things like that. So yeah, being in the deep ocean, you've probably got far less of a chance to. It's less visible than life on land to people. So it's a tricky one to raise up. Um, so a question here, so we'll kind of link to some of your past answers as well about after you collect your live samples, how long can you keep them alive for? And do you know how long their life cycle is? So this is very different depending on the type of bacteria that you get
For
on
sure.
Archaea
Yeah.
as well.
Yeah, it depends on what we're talking about. I tend to kill them on purpose because it's just kind of easier to get at the whole community if you can get the molecules out and the chemistry. Because what tends to happen when you grow them is that you get the usual suspects. You get these weeds, usually gamma proteobacteria, alpha proteobacteria, that are so good at taking up your whatever food you give them and growing that you lose the original community when you start growing things. So that's why I tend to kill them. So I preserve them and so I can look at how they are. But it's not impossible to grow some of these ones that are important in the environment. And so we just if you're thinking about the deep sea, of course, a lot of them die under depressurization. So you can mitigate that by putting them in a pressure vessel at the seafloor and then bringing back the closed pressure vessel. And so I have quite a few colleagues who've had good success doing that.
Are there some environments that it's easier to culture bacteria from and archaea as well, or is it obviously from I mean extreme environments, not as yeah, we can easily culture the ones we find on the table, but yeah, there's some that are kind of easier, or is that just how much we know about them so that we can kind of get the right conditions?
Yeah, I mean, it does matter how much we know about them and getting the conditions right. But I've actually looked at this quantitatively. We published this a few years ago to see which environments were the best to culture from, and our guts are the big winners. We're great at growing things from our guts, but which is good because we need to. But we're less good in other places. You do have a bit of an advantage when you're working with extremophiles because, as I said, you're throwing it in a vat of boiling water. So you're never going to get E. coli, taking over your culture and, a contaminant coming in. So it's actually, you do get some advantages to working with these extreme conditions. The really tough ones are the ones that use just organic matter, just sugars, and they're not hot and they're not high salt and they're not, anything weird and they're slow. And so then you just have to like fight everything, keep everything else at bay and let them grow.
Uh, Yeah, I think it's quite interesting, and especially because we think of kind of most of these things as quite fast growing, fast evolving, because we think a lot about disease causing bacteria. But then it's kind of indicating from this work, it seems like some of these could have been quite unchanged for a very, very long time, and very kind of ancient forms of life.
Yeah. And even some of the ones like the one that I showed the picture of, that's the most closely related single-celled organism to to the eukaryotes. That organism is even in culture, it's not in pure culture. It still needs some other cells around to support it. And also it does not grow fast. So a doubling time is, a doubling time is how long it takes a cell to divide into two, and then we get exponential growth. So you get like a lot of biomass just from normal E. coli that doubles every half hour or so. And so you can grow up a petri dish in a few hours. These cells that I showed double in weeks, like so 2 weeks, and then you just get the cells to divide, which I think is a lot faster than they are out in nature. So that's already speeding them up quite a bit. But it's like, you're never going to like run experiments and, be nimble and work with them in the same way that you can with something like E. coli.
Yeah, you might as well be working on vertebrates, so that
Yeah, pretty much.
counts.
Yeah, that's
Uh,
right.
yeah, no, it's quite it's very interesting, and it is just such an expansion of kind of what we know that's perhaps unparalleled in kind of the history of discovery of life, really, because I suppose most things were quite well known. I mean, it probably is kind of on a par with like discovery of bacteria in the first place. To kind of it's from what you're saying, it really appears to have broadened out the tree of life. And yeah, there's definitely can you think of the most unexpected place that you've ever found an extremophile? that you were just like, no, there can't be anything here,
Oh,
or things that you've read, or that colleagues have found.
yes, I actually have done exactly this. I didn't believe, and then I found, and then I discovered. There's a video, it's not very well watched, but we made a nice explainer video of early work that we did in Costa Rica with hot springs. And it's called Biology Meets Abduction. If you want to Google it, it's on YouTube. But I remember the filmmaker filming us and my colleague Donato Giovanelli explaining to the filmmaker as I'm in the background, and he's saying, what we're doing here is that we're filtering the water and we're catching the cells in these sterile filters, and then we'll take the cells home and we'll study them. And as I was listening to him say this to the camera, I thought, this is clear water. It does not look like there's anything growing in it, and it is like 90 degrees Celsius. And I was just like, how did he say that with such confidence? And I was absolutely proved wrong. We got those, he was right. We got those filters back to our laboratories and we found lots of cells and highly diverse ecosystems and definitely related to the subsurface. And so that we've been expanding on that work ever since. And I don't doubt that anymore. But at the time, I had never worked on those kinds of samples before, and seeing that you know, clear fluid just doesn't scream biology to me.
It's quite an unusual find. Just yeah, hard to believe. Um, yeah, so a lot of these a lot of these new discoveries seem to be finding species that are kind of really really proficient in lots of unusual types of chemistry. Do you think there's a lot that we can learn from some of them, and processes that we didn't even imagine could be done?
Yeah. One of the great motivations for one of my colleagues, Jan Amund, who's since passed away, but he was absolutely driven by writing out those equations on a piece of paper, looking at them, doing the calculations, and saying that is an equation that should support life. And nobody's ever found life that uses it, but I'm going to look for it. And so his most recent through his student Heidi Aronson has now published this, but they found Kamamox. So this is taking two different oxidation states of sulfur and then basically combining them to meet in the middle and end up with something, an intermediate oxidation state. But that's not a metabolism, an energy metabolism that anybody had ever known before. And he predicted it just simply based on the chemistry. And then they found it and proved that it existed.
Pretty good to be validated. So we got a couple of more question. The question of if the deep sea crab ever got named. I presume as a species, although it might be
Oh, the one
asking
in
if the
my
individual crab got a name. I don't know.
I don't study animals. I like looking at
Too big
them.
for you, that one. Yeah.
So big. It's like more than one cell. It's way too big. So I don't want to answer that because I'm not the expert in that. But I
I,
do.
I think they have been described, but I don't know the name of those.
Many animals in those places are often not described. So it's not crazy to discover a new species of animal down there. But that's, I have colleagues who do that kind of stuff, and I don't want to speak wrong.
Yeah. Someone will have described them now. There's footage at somewhere, right? But it's
Yeah.
yeah,
Yeah.
we don't know. um,
But it's funny because even those of us who don't study these animals, we're still entranced by them. Same thing with dolphins. You can be out at sea, you can be this hardened oceanographer, but if somebody's like dolphins off the starboard bow, everybody rushes. It's cute.
So, just got a question asking about if you can comment on extreme life in cold places. I mean, so you mentioned you working a bit with some colleagues at the British Antarctic Survey. So yeah,
It's amazing how much life we find in frozen soil. Like you'd think that there'd be nothing. But it actually, the way ice forms, so ice is pure water forming a crystalline, obviously it's ice. We all know what that is. But when you talk about like the water that's sort of saturating soil, when you pull out fresh water and make ice with it, as it freezes in the fall, the leftover wet stuff gets a higher and higher concentration of any salts that were present. And so as you continue to pull ice out and pull ice out, you get this more and more concentrated until it turns into a brine. And then its melting point has dropped so low that it doesn't melt. And so you actually find tiny, tiny little brine veins, even in ancient permafrost, that can support life. And so the life that we find in these brine veins in the middle of winter in fully frozen soil is, I think, fascinating. We can do tests to see if they're functional, to see if they're alive, and they are alive. So there are things that can basically survive off this tiny amount of brine fluid, even while it's frozen.
Yeah, I found reading about your work with the permafrost quite interesting just because there's so many unknowns. We don't know what's going to happen when it starts thawing out to these communities, which obviously has quite a big impact. So it's kind of worrying, but then there's also a hopeful tint that it might not be as bad as some people thought. There might be positive outcomes there from all the kind of um, yeah, there's the methane that could be released or locked up, but we don't know because they're so poorly studied the communities there. Obviously, you and colleagues are gaining information all the time, but yeah, still pretty hard to work
Yeah,
out.
we're just like the tip of the iceberg, which is that a pun or there's icebergs and they're beautiful. But it's when you work in the Arctic, there's no question that climate change is happening and it's happening rapidly. You see it happening before your eyes. So this is a real, it is happening. But we're there for it. We need to continue. We need to, the answer is not just like shutting our eyes and ears and staying away and saying, well, let's give up on that place. The answer is to study it and to learn as much as we can.
I think it is very interesting that it wasn't too long ago that people thought any like the search for new life was mainly focused on other planets, and then the hydrothermal vents kind of changed that. So it's yeah, it's very interesting how things can just change so rapidly, and there's so many parts of our own planet that we just don't know about. So yeah, really interesting
Yeah.
to hear about your discoveries. That leads me to yeah, to thank you for such a fascinating talk, and thank you as well for answering all these questions from the audience members. And yeah, and I'd just like to thank the audience so much for attending today's event, and be sure that they check out what else is coming up in the Festival of Ideas program.